Okay. So just as a quick reminder, we had two things due today. The first was a pre-lab worksheet, but the other thing that was due today was the enzyme action, the catalase full paper. I had that due for 5 p.m. I think officially in the Dropbox. But so if you have any questions, we can I can help you out a little bit with any questions you might have during lab today. When you finish up with the lab activities for today, we can go through the paper there a little bit then as well. But it is due today. I'd rather have a good product than a rushed product, though. Ideally, you're getting it in today, but if you have any questions, please ask those. If you feel like it does need a little bit more time, if you get it turned in sometime tomorrow, that would be fine with me then as well. Okay, but I do want those in this week if possible at the absolute latest. Remember that assignment is required in order to pass this course. So it is a required assignment. Otherwise, today what we're going to do is start investigating cellular respiration. And this is going to be also looking at a little bit of the enzymes that are inside of cells, but that enzymes often have substrate specificity. That enzymes will only bind to very specific substrates and then speed up chemical reactions related to those specific substrates or reactants. I use those two terms, reactants and substrates, pretty interchangeably, just to let you guys know. So we're going to investigate substrate specificity when it comes to enzymes. And really what that does is, because we're investigating both of those kind of put together, we're determining what types of sugars yeast, and specifically the enzymes in yeast, can recognize, can process metabolically, and can and therefore if they can recognize those sugars, if those enzymes that the yeast have can bind those sugars, then they're going to also break down those sugars via cellular respiration, using them as an energy source. In essence, we're looking at what food sources, what sugars can yeast eat. And no, not all the sugars you guys are using today are ones that yeast can recognize and use. So there's at least one sugar out there that you guys should probably be able to see that yeast doesn't have the enzymes to properly process it, and it really does not have a big impact on respiration rates. Okay, so that's one of the things you're looking for today. Again, yeast, we talked about yeast already previously. It's a single-celled fungal organism, and it's one of the simplest eukaryotes. So, these are some slides I went through previously, so I'm just going to go through it. Yeast is really heavily researched, though. Yeast is very easy to grow, and it's very cheap to grow, and it grows very, very fast. Those three things make yeast a relatively simple organism, cheap organism to do experiments on. And the nice thing is it also has a fully sequenced genome. So we know what its DNA sequences are all along its chromosome. We know a lot of what those genes do. We know how to manipulate those genes genetically. They can knock out a gene. One of the ways you can actually figure out what does a gene do in an organism, say you're studying yeast, you want to figure out what gene X does. The simplest way to do that is you delete the gene from the organism, and then you see what goes wrong. So if you delete a gene, what's it called, a gene knockout, and then you see what becomes defective, you got a pretty good idea what that gene does and the protein it codes for does that. So that's one of the simpler ways to do some of these things. You can manipulate these genetically, see what goes wrong in essence. Yeah, again, another nice thing about yeast is a good approximation of human cells. There's several processes like mitosis, meiosis, that take place in yeast cells and are almost identical in human cells. So we can use these as something easy to study, cheap to study, unlike, say, human cells, which are very difficult to study, can take a lot of time, and have some ethical constraints then as well. And besides that, yeast also has a number of commercial purposes. So knowing about how it grows and how it functions is important for things like making bread or alcohol, or a number of pharmaceutical applications then as well. And even in the area, anybody ever heard of Red Star Yeast? It's a big company in our local area. And it does, yeah, a lot of yeast production, like the yeast you buy in the store to make bread, the dry activated yeast, but actually they make a lot of yeast for research purposes as well. So it's partially a food company, it's partially a biotech company, for instance. All right, when it comes to what we're looking at today is you might have, this is supposed to be my yeast cell. It's floating around, say in some type of solution. We're gonna be working with yeast a little bit later, but the yeast, if it has a food source, a lot of the nutrients that a yeast cell might like to eat floating around in the environment around it are in the forms of the macromolecules. Do you guys remember what the 4 different kinds of macromolecule classes are? What are the 4 different kinds of macromolecular classes? What? Lipids and fats, proteins, carbohydrate, sugars, and nucleic acids. DNA and RNA fall into the nucleic acid category. So again, we got proteins, lipids, carbs, carbohydrates, and nucleic acids. So a really good source of nutrients is things like a polysaccharide carbohydrate. Polysaccharide, what's poly mean? Many. Saccharides just means sugar. So what this is 1, 2, 3, 4, 5, 6 sugars joined together, a lot of sugars joined together in this polysaccharide chain, many sugars joined together. And that could just be something like glucose, 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 for instance. It could just be many glucoses joined together. So you might have a polysaccharide sugar floating around in the environment. You might have a protein floating around the environment. What are proteins made out of? What do you put together to build a protein? It is a type of acid. Amino acids. So you build a protein by putting many amino acids. How many amino acids are there? Nope. 20. There's 20 total amino acids. I don't know. Is that a lot?
That's quite
okay. That's a lot. So you have these different amino acids get put together. So a lot of these nutrients are sequestered in the form of the macromolecules, floating around in the form of macromolecules, like a polysaccharide sugar or protein. These are really good carbon sources, nitrogen sources, sometimes hydrogen sources, oxygen sources, just energy sources. The cell would like to get some of these nutrients. So there's a lot of energy. Macromolecules tend to be energy-rich, nutrient-rich molecules for cells. And that's good. The problem is they're usually too big to transport and fit across the membrane. So an entire protein cannot pass freely through membranes. Remember, membranes are selectively permeable barriers. So, and even if they had like a nutrient transport channel or a few other things, a lot of times an entire protein molecule or an entire polysaccharide molecule is just simply too big to get into the cell very easily or hardly at all. So cells have to have some other ways to get nutrients in. And usually with something like yeast, what they do is they secrete out digestive enzymes. So they make enzymes that digest other molecules. So for instance, proteases, these are enzymes that digest proteins. They are nucleases, these are enzymes that digest nucleic acids. There's amylases, these are enzymes that digest polysaccharide sugars. There's lipases that digest lipids. What happens is something like a yeast cell might make these enzymes and secrete them out to the environment. So something like my amylase enzyme might come over here and it lands and hits this polysaccharide. Something like my protease might come over here and hit this protein. And then they're going to digest them apart. They're going to tear them into smaller bits. And if you're digesting a polysaccharide, you're basically maybe digesting it down to monosaccharide sugars, like an individual glucose molecule. Maybe you're taking this protein and you're digesting it apart into smaller bits. And again, smaller bits of a protein is amino acids. So maybe they're digesting these proteins, big protein molecules, into individual smaller amino acids. Now, this is then small enough to bring into the cell and use as a nutrient source. And this is what is called sometimes nutrient mining, but probably more properly extracellular digestion, where they secrete out these digestive enzymes. They start the digestive process outside the cell, then they bring in the small digested products like amino acids or monosaccharide sugars through some transport channels, nutrient transport channels in the membrane. And this is how fungi feed. This is how a lot of different bacterial species feed. Technically, this is how we feed. What do we do when we eat food? Does the food particles just go directly into our cells? No, it goes to our stomach and our intestines, our digestive tract. And what do? We secrete a bunch of acids and digestive enzymes into our stomach. And then into our intestinal tract, they break down the food into little tiny small bits that then pass from our intestines to our circulatory system, and then from our circulatory system to all the cells in our body. But they got to digest it into the little tiny small stuff first. Does that make sense already? So all this is different types of extracellular digestion that starts on the outside of the cell. But they got to have the right enzymes. They got to have the right enzymes for breaking these down. They got to have the right enzymes for transporting them in the first place. These transport channels, this might be a transport channel that is specifically for transporting amino acids. This might be a transport channel for specifically bringing in glucose and just glucose. So a lot of these nutrient transporters are substrate specific. They specialize in bringing in one kind of nutrient for the cell. Now with cellular respiration, then once the stuff gets inside the cell, it can go into a process, metabolic process like cellular respiration. And with cellular respiration, you can have sugars, carbohydrate sugars can go into cellular respiration, fats and lipids can go into cellular respiration, proteins or amino acids can go into cellular respiration. Those are all things that can go into cellular respiration and get consumed to fuel the cell as energy sources for the cell. And the overall purpose of cellular respiration, energy processing. Cellular respiration is energy processing reactions for the cell, generating energy in the form of what are called energy intermediates for the cell to use and do work. So it's a way of getting energy and processing energy. Typically then, have you guys started talking about cellular respiration, those in Bio 1 lecture? Okay. So these are all things that can go in, but usually when we teach it in class, we just say glucose goes in. We're just simplifying it and say C6H12O6 glucose. Sugar goes in, but any of this other stuff could potentially go in to some of this as well. But just to simplify things, we just talk about glucose catabolism. And with cellular respiration, and by the way, you guys have a lab quiz next week. So lab quiz number 3 is next week on the 1st. And I might say, oh, here's the reaction, the overall summary equation for cellular respiration. Yeah, you probably need to know that for the quiz. Why? Because you probably need to know that also for the lecture, for those of you that are in Bio 1 lecture as well. So my hint is memorize this. You do need to know that. I will ask you on the quiz next week. So the overall summary equation of cellular respiration is C6H12O6, glucose, plus 6 molecules of oxygen gas, so 6O2. These are going to be your substrates or reactants. They are going to go through a chemical reaction that then is going to give rise to the products of 6 carbon dioxide molecules, 6 CO2 molecules, 6 water molecules, and then energy is going to get released from this. Some of that energy is going to get tied up by the cell to make ATP, adenosine triphosphate, but some of that energy is also going to get lost as heat energy to the surroundings. Chemical reactions are never 100% efficient, so some energy is always lost as heat to the surroundings. Now, this is the overall summary equation of cellular respiration, and it looks fairly simple, except for cellular respiration is not fairly simple. There's a lot of different steps involved in this process. And this is just what happens from the very beginning to, in essence, the very end. But there's a lot of stuff that happens in the middle. There's all this. This is an overview of cellular respiration. Cellular respiration consists of 3 different main metabolic pathways. You guys know what a metabolic pathway is? We talk about metabolism is all of the chemical reactions inside of the cell. What do you think a more specific metabolic pathway is?
The way one reaction happens.
So, sorry, what? So it is about the way reactions happen in the cell, but it's a little bit more specific than that even. A metabolic pathway, an example of a metabolic pathway is something like glycolysis. You guys talked about glycolysis in lecture already, those in Bio 1 lecture? Is glycolysis just a single chemical reaction? No, it's a series of interlinked chemical reaction. What is a metabolic pathway? It's a series of interlinked chemical reactions. And what I mean by that is the product of chemical reaction number 1 becomes the substrate for chemical reaction number 2. The product of chemical reaction number 2 becomes the substrate for chemical reaction number 3. So it's a series of this chemical reaction leads to this one, to this one, to this one, to this one, but they all have a common function. So something like glycolysis, this is a metabolic pathway, a series of interlinked chemical reactions. But glycolysis specifically breaks down glucose to generate ATP and NADH for the cell to use. So the function of glycolysis, break down glucose to get energy for the cell. So typically cellular respiration is glycolysis, the citric acid cycle, and something called oxidative phosphorylation. Those are the 3 main pathways, metabolic pathways, that are all part of cellular respiration. Glycolysis, glucose goes in, and what do you get out? You break that glucose down into pyruvates, 2 ATP molecules, and 2 NADH molecules. So again, glycolysis, overall, glucose goes in, you get 2 NADH, 2 ATP. These are what are called energy intermediates, the way the cells store energy, and then they can use those up to do cellular work to accomplish something. And you get 2 pyruvates. The pyruvates are going to go into the citric acid cycle. They get broken down even further. And when you get out of the citric acid cycle, you get a couple more ATP, you get some more NADH made. And then also those pyruvates are 3 carbon molecules, and they get broken down into 3 separate CO2 molecules. So remember, glucose is C6H12O6, 6 carbons in glucose. Here it gets broken down into 2 pyruvates. So there's still 6 carbons. You guys see that? They're kind of following along with that. And then those pyruvates both go to citric acid cycle, and those 2 3-carbon pyruvates, again, that's 6 carbons total, then also are going to generate 6 different CO2 molecules over the course of the citric acid cycle. In essence, again, 6 carbons. Okay, so we balance that equation out. This makes some more ATP, it makes some NADH, and then the NADH from glycolysis, the NADH from citric acid cycle are going to feed into oxidative phosphorylation. This is the electron transport chain, which takes high energy electrons from NADH. And what does the electron transport chain then make? Anybody happen to remember that one?
What was the question?
What does electron transport chains make? NADH goes in, high energy electrons from NADH goes into an electron transport chain. The electron transport chain is going to use the energy from the electrons to do something. And what's it NADH2? And nope. It's not ATP. It's not ATP. It uses it to put something on one side of a membrane. Have you guys talked about that in lecture yet? Am I ahead? I don't know where you guys are at in lecture.
We just were on part of the ADP and.
So what it's going to do is it pumps protons, H+ ions, to one side of a membrane in the mitochondria. Okay, so electron transport chains make a proton gradient across a membrane, or sometimes what is called a proton motive force. And then you also have chemiosmosis as part of this process. Chemiosmosis is use of an enzyme called ATP synthetase or ATP synthase. ATP synthase will consume the proton gradients. The proton motive force is the energy source to make ATP. So this doesn't make ATP, it makes a proton gradient across the membrane. This consumes the proton gradient across the membrane to make ATP. So there's 2 different parts to oxidative phosphorylation. Questions about that? By the way, a great way to study for lecture is to do what's called a concept map. Anybody ever done one of these before?
It's like a frame.
It is, but draw out what goes in and what comes out of glycolysis. And then the stuff in glycolysis, where does it go next? So for instance, you can draw out a cell, put a mitochondria in the cell. Mitochondria is double membrane, has an inner membrane, and then it has the folded inner membrane. What are the membrane folds called of a mitochondria? Anybody remember? Christe? And so what's going to end up happening, so is you can draw out where these processes are located, so glycolysis in the cytoplasm. Citric acid cycle is in the mitochondrial matrix in the middle of the mitochondria. Electron transport chains are enzymes that are embedded inside of the inner membrane of the mitochondria, and they pump protons between the two membranes to the inner membrane space. For instance, you can draw out the relative locations of these. You can draw out what goes in, what comes out of each one of the metabolic pathways. You can draw out also then how they connect together. So the NADH from glycolysis, that's made in glycolysis, feeds into electron transport chains, for instance. You can draw this out on a single piece of paper, and you can take all that stuff that was probably covered in 2 lectures and have one study guide. So that's a really useful process, but you need to draw that out for yourself and plan it out for yourself, because that's actually the process of making that chart helps you remember it all as well. All right, today's lab, we're going to investigate cellular respiration, but we're going to take a look and investigate specifically how different kinds of then sugars are able to be used by yeast or not used by yeast to do cellular respiration then. So we're specifically looking at the rate of respiration in the presence of yeast mixed with either glucose sugar, yeast mixed with sucrose, yeast mixed with fructose sugars, yeast mixed with lactose sugars, and then our control is just going to be yeast mixed with water, no sugar at all. Okay, and there will be a little bit of respiration even in the water control. Sometimes when the yeast doesn't have anything to eat, they start cannibalizing their proteins. They break apart their proteins, eat them as a source of amino acids to keep their respiration, their energy processing going. That only lasts for a while, but it's long enough that we're still see some background rate of respiration. That's going to be considered our kind of not respiring control. Okay, you'll still see a little bit, but that's what it is. Questions about that? All right, what we're going to do is see, can the yeast eat glucose and how well? Can they eat sucrose and how well? Can they eat fructose and how well? Can they eat lactose and how well? And how well they do it, the better they recognize and process a particular sugar, the higher their respiration rates will be. But there's probably going to be at least one of these sugars they cannot recognize, and its rate of respiration is probably going to be similar to the water sample. Questions about that? All right, we're going to get this started. Okay, so why might a yeast not process a certain type of sugar then? What will make a yeast unable to process a sugar?
Doesn't have like the proper digestive enzyme.
Yeah, remember, enzymes are very substrate specific. So that I brought up at the very beginning of today. If the enzyme cannot recognize and bind to a certain sugar, the yeast or any other cell that it doesn't lack that enzyme cannot process it. So there might be one of these types of sugars that is one that yeast cannot recognize because it doesn't have any of the enzymes to bind it and recognize it. Could be that it doesn't have a transport enzyme to bring it in. Could be it doesn't have the right enzymes for it to feed into cellular respiration, for instance. So they're just not going to have enzymes that recognize this. Okay. All right, cellular respiration, we're looking at today. Yeah, again, this is the overall summary rate. How would you track the rate of this reaction? What are we going to be looking at today? Yeah, we're going to be looking at CO2 release. So if you guys look at your sensors out today, they're CO2 sensors. So just like we've been tracking oxygen percentage previously, now we're going to be tracking carbon dioxide production over time. We're looking at the appearance of one of the products over time from this process. Okay. In essence, what you're going to do is, and I think some people have already started this from what I've been watching, you're going to label 5 different test tubes. One of them is going to be labeled for glucose, 1 sucrose, 1 fructose, 1 lactose, 1 water. And you can just write GSFLW if you want at the tops of the tubes. You're going to place 2 mils of the appropriate sugar solution in each tube. You have those sitting out in the middle in the conical tubes. You should have the sugar solutions. You're then also going to place 2 mils of a yeast solution into each tube. When you guys have those little plastic pasture pipette things, just to let you know, you probably haven't been paying too close of attention to it, but there's a little marking on here that says 1 mil, and it's this last marking before you hit the bulb. So if you fill the pipette up from the tip to here, that is 1 milliliter. Okay, so that's how you're going to measure out the milliliters. Just do so fairly carefully. Try to be as consistent as possible. You're going to put all of those tubes for 10 minutes at 32 degrees Celsius. So this is my water bath over here. Looks like it's running maybe a little hot at the moment, but it's fine. And so with that one, you're going to put, actually, no, it's fine. All right, so you're going to put them in there for 32 minutes, 32 degrees Celsius. You're going to keep it there for 10 minutes or so. Prep up your computer programs. And after the incubation, you're going to go and start assaying the rate very similarly to what you have done in the previous lab. The veneer programs, now with the CO2 sensors, the Nalgene bottles. What you're just going to do is after the incubation period, you're going to mix it. Make sure you mix it. You might have to invert the tube real quick. You got the little parafilm pieces out there to help you with the inversion. But then you're going to hook out, don't dump the entire thing into the Nalgene bottles. You're going to take out 1 mil out of your sugar yeast solution that had been incubating for 10 minutes, put it into the Nalgene bottle, put the CO2 sensor in, take the readings, okay? And then determine the rate of reaction, similar to what you guys have been doing. Make sure you're also going in the right order. Do glucose, then sucrose, then lactose, and then finally do, so go in the order of glucose first, then sucrose, then fructose, then lactose, and water at the end. Please go in that order. It just helps the experiment work a little bit more properly. Questions about that? All right, assignments. Real quick reminder for assignments. Pre-lab worksheet number 7 is due next week. You're going to have a statistics assignment next week. And yes, that does mean I need to talk about statistics yet. So we'll do that before we get started. And you do have a lab quiz next week, lab quiz number 3 as well. Question about any of those? Yeah, pre-lab worksheet number 7, and you have a first statistics assignment. Yes, that does mean there'll be a second one here later.